Good evening, everyone. I hope you guys are having a good week. So, for our second lecture, we're doing chapter 1.2, the scientific method. And again, this is chapter 1, unit 1. And I think unit 1 is going to be changed to cells. Because right now we have it as ecology, but it's going to be changed to cells. So, our aim for today, instead of putting objective under the title, I'm going to put aim. Aim. What are the steps of the scientific method, and how can we use, let's change that. How can we use the scientific method to create an experiment? So that's our aim for today. So what is science? What is science and is not? So science is a way of using evidence to learn about the natural world around us. So that's how we learned almost everything in science. Someone saw something happen, they thought about it, experimented on it, had results, did this multiple times, and then we came up, when that same results kept repeating and happening a lot, they created explanations for phenomena that happen in the world. So the goal of science is to investigate and understand the natural world, and to explain events in this natural world, and use those explanations to make useful predictions. Not just regular predictions, but useful ones. So the scientific method is a series of problem-solving procedures that might include observations, forming hypotheses, experimenting, gathering, and analyzing data. Sorry, I should have put "and" analyzing. That's a comma. Analyzing data and drawing conclusions. So the first thing of the scientific method is to 1. Make an observation. So observation is just the gathering of information or processes in an orderly, careful way. So the first thing we do, we just see something. But I can't be like, I see a yellow wall. It has to be something that's useful, something that we observe in our natural world. Like a question's like, why do giraffes have long neck or instead of short necks? Why do, like my puppy, when she eliminates waste, she watches me intently. And it's not the first puppy I have that I've seen other puppies do it, or dogs do it. They watch their owners or whoever that they trust intently as they're defecating or eliminating their waste. So the information gathered from observation is called data. So there are 2 types of data. There's quantitative and qualitative. Quantitative, think N. Quantitative is a number, or something you can measure, counting. Qualitative is a description or involves characteristics that can't usually be counted. It's something that's not a number. So some examples. Qualitative. So she's measuring something that involves a number. So this is a quantitative thing. And then this is something that's not a number. Like I can't say that fish looks like a number 2. That's unuseful data. It's orange and it's adorable. It looks like Nemo. So it's a qualitative. So after you make that observation, the first thing you do, come up with a problem or a question based on that observation. So develop a question or problem that can be solved through experimentation. So it has to be solved through some type of experimentation. That's very important. Next, you're going to formulate this cool thing called a hypothesis. A hypothesis is a testable explanation. And again, it must be testable. So it has to be in an if-then statement. So example would be this: if soil temperature rises, then plant growth will increase. So that's a testable if-then hypothesis. The example will be example of a bad hypothesis. I can't remember. Let me think. If I turn my textbook, then I'll be on. You could technically test that. If I turn my textbook, then I'll be on the next page. Like, I don't know. I'll think of one eventually. Let's just keep going. So, experimental design. Wait. Let me make sure. We seem to have skipped. Oh, yeah, okay, we're fine. So, after we formulate this hypothesis, we're gonna test it. We're gonna perform an experiment. So, there's certain criterias for experimental design. So, in order for to have a good experiment, it must be replicated, not like once or twice. So sometimes, so this is what we do results. We do experiments as a class, and in class, like for our lab reports, we're not gonna just use our data. We're gonna use class data. So, because we don't want to like do the experiment like 3-4 times over, because it's gonna take forever. But in order for experiments to be good, so you have like valid results, it has to be replicated. And then the validity of experimental results must be tested statistically. So I don't know if we get into statistical testing. If you're majoring in any science, especially biology or chemistry, you have to take statistics because you're consistently using it. So statistic is just a numerical way of saying whether a result is valid or if the information goes for or against your hypothesis, if it's not just due to just randomness. So here are the key common things in experiments. Experiments, they have to be controlled. Reason why we want to control that, we need something to test our results to. So in a controlled experiment, there are two groups. There's an experimental group, the group that's being altered, and the controlled group, where the variables are not being altered. So let's say I want to test if caffeine affects mice, right? So if I just give all my mice caffeine, like I don't know how they would normally act without the caffeine. So I'm going to have a group of mice with, one type of coffee and another type of coffee, and that's going to be my experimental group. And my control group is just going to be a group of mice with just water. So in experiments, they use models to recreate occurrence, but in a controlled setting. So what that means is that when you design an experiment, you only want to change one thing, because that's going to make our results more detailed. It's going to make it more accurate. If we change a bunch of other stuff, we're not going to know what is attributing to the results that we're getting. There's too many factors. So to make results as effective, as clear and coherent and precise, we want to only change one thing, and we want to keep every little thing the exact same. So again, in that mice example, like I want to keep the temperature when they can drink the water, how much the water, and not just the water but the coffee that they drink. I want to keep the environment the same, basically everything the same. So how do we design a controlled experiment? So the factors in the experiment that can be changed are called variables. So things a lot. So in lab and mostly lab and life, things that we can change. We can temperature, light, if you're doing a plant, the time for the mouse, the concentration of the solution used, the external environment, and not just time, but the time of day, the amounts of things. So that in a controlled experiment, a controlled experiment works again with only one variable. When you work with one variable at a time, several variables were changed. At the same time, science would know which variable was responsible for the observed results. So continued. So in a controlled experiment, again, only one is changed at a time. All variables should remain unchanged or controlled. And there should be a controlled group and an experimental group. And the two groups are identical except for one factor. The control group serves, it's the comparison. It's like what I'm basing everything off of. If we don't have a control group, we have nothing to base. There's no baseline. There's no norm or normal occurrence or results. Okay. So example, in order to test the effectiveness of a new vaccine, 50 volunteers are selected and divided into two groups, the controlled and the experimental. Both groups are given given a pill to take that is identical in size, shape, and color. So clearly the one is a vaccine and one is not. So the first part would be to describe the control group. So even though they're volunteers, the control group will have one that doesn't have the vaccine, and the experimental group will have the one that has the vaccine. So which variables are kept the same? So it could be like the time of day, the size, shape, and color of the pill. And to make this even more like accurate, you want to do the same time of day, because there are certain things to attribute to when people take things. And then variables that are being changed in this person is whether the pill has a vaccine or not. And only one thing being changed. So let's go more in depth into variables. So a variable, again, is a factor, a trait, or condition that exists in differing amounts of types. So there are three types of variables: independent variable, dependent variable, and a controlled variable. So an independent variable, independent. So this is the one that is changed by science. It is independent. It doesn't matter the results of the experiment. There's nothing depending on its results. So as a scientist change the independent variable, he or she observes what happened. So okay, and then the scientist focuses on observes on the dependent variable. Those are the factors that are being measured and controlled. The things that we depend on. So like examples of dependent variables could be like time, because in that mouse experiment, like the time, the experiment is dependent on like the time, or the amount of caffeine would be the dependent. The amount of caffeine would be the independent, and the time of day would be dependent. As we go through examples, we'll point out the independent and dependent variables. So in the experiment, there's also a controlled variable. Those are the quantities that remain consistent, and they must observe them as carefully as the dependent. So after we do this elaborate whatever, we're recording and analyzing our results. So the data that has been collected must be organized and analyzed to determine whether the data is reliable. That's where our statistics come in too. And then we have to decide, when we're drawing that conclusion, does the data support or not support our hypothesis. So let's go further into data analysis. So data analysis, you want to keep careful and accurate results of what you see or the data you collect. And then you have data analysis. That's where the math and statistics are used to determine whether an observation or experimental result is meaningful or just random or happened by chance, by coincidence. That's why statistics is very important. Researchers work on complex questions, often in collaborative teams, reviewing, critiquing each other's data and hypotheses. This helps ensure that the conclusions are valid. So when you're doing research, you have a million people looking at your results, and not only just that, like you have this big question, this overarching question, and it's broken down into a bunch of different little experiments, and those experiments are repeated over and over and over and over. So when I did my undergrad research, we were looking at a genetic disease, and I was literally looking at one of the cells. My friend who's also in my research group was looking at another type of the cell. So I had the epsilon cell. She had the I don't even know what cells she had because I forgot. But she definitely had the easier cell. I had the newer, not much information tested about it type of cell. And that's just answering one little thing about this one genetic disease. But not only that, every day I had a petri dish. I had more than one. I had 25 to 30 petri dishes, and I would perform the experiments on those same petri dishes. So I actually had 50. So 25, I would start one trial of the experiment, and the other ones I would grow to have more to do my next trial. So I did about one trial every 3 months, that whole entire process in a like 3 to 4 or 5 month span. And I would do that over and over and over and over on thousands of cells. And like that's not even enough because cells are so small anyway, and half of them, like more than half of them, would die. But again, we would, and like I was the only person doing that. It was just you have to like consistently do experimentation. So your data, everybody's data looks different. So we'll go through some types of data. So like for in your data section, like when you're doing lab reports, like you should always have some type of graph, or like not even, I can't say picture because a picture doesn't tell me anything. Even though picture stuff, it tells me nothing about your data. So this is one type of data, and this person did the number of adults and children living with HIV in 2008. So this is a type of data, and it's a type of way that this person represented that data. And then organizing data into graphic illustrations helps scientists analyze the data and explain it clearly to others. So it's not annoying, but it's, even while we're in class, to hear like, oh, I don't want to read, I don't want to read. That's the majority of the world, people. But that's something we have to learn to appreciate, is reading. And the only way we do it, reading, by actually reading. But graphs and data, it's just a quicker way to see someone's results, and you can make good tables are a good way to just summarize the results as opposed to reading 3 paragraphs, just only when you want to just quickly glance at it. Here's another way: the leading cause of death amongst adolescents 15 to 19. We see, what is this? Unintentional injury. Interesting. Another graph. I don't know why this is cut off. This is another type of graph. This graph is really useful when you're comparing two quantities. So like this one is comparing Canada to First Nations, and these are a bunch of different categories. So age, it's our independent variable, and the rate per thousand population is a dependent variable. So guys, in your notes, draw a graph and note that the y-axis over here is always the independent is it dependent? Yeah, this is always the independent variable. And here at the x-axis is always the dependent variable. Always, always, always. That's how you want to display information. Here's another graph. Again, this is the independent variable, so the numbers of scoops. These are the things that's changing. And then the dependent variables, these are things that are not changing. So the participants are not changing, but the numbers of scoops are changing. And there's another pie chart. There's another way to represent data. So after we are done doing our experiments, we have to draw a conclusion. What is the evidence? What evidence from the experiment is used to determine whether this hypothesis is proven or disproven or correct or not correct, supported or not supported? This is when, again, that also comes in our data analysis. We based on our data analysis, our chi-square that we had to, this is a thing called chi-square. Based on that number, it tells you whether your experiment was just happen by the results happen by chance, or there's something significant to it. So again, experiments must be repeated over and over when results should always be the same before a valid conclusion can be reached. So if you have wavering results, you don't have a valid conclusion. So to help people in the world, and especially people who are furthering results based their own results, and your experiment can help them, you must always, as a scientist of the world, communicate those results, because other people are interested in what you're studying and what you're doing. So you have to publish the results in a scientific article. Must tell the reader what the question was, and not only what the question was, what the question being answered, and your hypothesis. Why the question is important, relevance, some background information, and precise description of how your work was done. That's your procedure, the data that was collected, and the scientific evaluation of what that means. So you don't publish results only when it supports your hypothesis. You publish when it does and doesn't, because your mistakes, people learn from mistakes, not from perfection. If you already know something, you already know it. You didn't learn anything new. You knew it already. You understood it. The bulk of learning always comes from error. So if you want to take away a life lesson, take away that. Was mistakes? Good. Very, very good. So again, data is shared with the scientific community through research articles published in scientific journals. I know one of the scientific journals I'm subscribed to is what is it? It's Cell. That's the scientific journal. And then the medical journal of what is that place? I can't remember. But there are loads and loads of scientific journals, and they could be published as research papers, peer reviews, and then yeah. And also, if you're collaborating with someone, you can connect with them via phone or email, or now that our technology is so advanced. For me, I like Skype because, I don't know, face-to-face contact is much better. So after you get these results, let me see. Okay. So if a hypothesis is supported in ample experimental data, not just your experimental data, I'm talking like a bunch of people repeating, it leads to a theory. A theory may be defined in a more, maybe defined as widely acceptable, plausible general statements about a fundamental principle in science. So example be the germ theory states that infectious disease are caused by microorganisms. So it's very broad, but it says that infections are caused by it's like many diseases are not caused by microbes, so we must be careful not to generalize theories too broadly. So I can't just say again, so it's like microorganism causes all infectious diseases. That's not true. And then a theory continues to be tested and modifications are made as we learn more and more and more about the theory. So everything starts right there. And then once a theory is consistent, it becomes a law. So if you were in my chem class, you guys would have learned about the laws of conservation of mass. So law of conservation of mass is saying that no matter what, like a group of elements in the product side is going to stay the same reactor side. It's just saying that mass is conserved throughout a change and experiment. And the reason why they learned it because this guy Jake Dalton. But again, this is a lot. This is something that's been tested over and over and over and over and over and over. Has been made modifications, and if there are more modifications to be made, they will be made. But this is there's no more modifications to be made. This is a lot. This is something that will happen over and over and over again. So another example with this, so we're going to do cell theory. Again, it's a theory. But this is the first rule in cell theory. All living things come from pre-existing living things. So that's just a law. Like, that doesn't happen. Like, my dog's food can't turn into ants. Or back in the day, they believed that if you left rice out, it would transform into mice. That wasn't true. They didn't know that the mice were eating through the bag, and then when they opened it, they would find mice in the rice bags, and they just thought like, oh no, like mice came from rice. Maybe that's why they rhyme. Okay, and scientific law promotes the process of generalization. Once a theory becomes established, it can be used to predict specific events. Okay. Again, guys, if you need to go back to clarify stuff, go back and clarify. Let me go through that slide. So this is the end. Remember to submit the 2 questions you have about the lecture. Your questions help me help drive the next day's instruction, or for the lesson. Like, what do I need to reteach or clarify? So I read your questions. They pop up on my phone as text. So, and then reminder quiz on Friday. So the lecture for Thursday night, at the end of that lecture, I'll do a quiz review. Okay, guys, have a great day. I'm gonna end this.